Bismillahirrahmanirrahim. Today the topic of my discussion is Kreb cycle, that is also called as TCA cycle. Kreb cycle is also known as tricarboxylic acid cycle, and it is a biochemical pathway that is used to generate energy through oxidation of acetyl CoA. It is used for the synthesis of NADH and for the production of amino acid. It was first described by Sir Hans Krebs in 1937 and got Nobel Prize in 1953. This cycle is called as Kreb cycle, and it was later named as citric acid cycle that was discovered by Sir Augustin. Kreb cycle occurs in mitochondria of eukaryotes and in the cytosol of prokaryotes. And we know that what is the difference between the prokaryotes and eukaryotes. Mostly the simple microscopic organisms that are called as bacteria are prokaryotic, and the rest of all the kingdoms contain mostly the animal kingdom, plant kingdom, fungi. They are all eukaryotic organisms. Krebs cycle is an oxidative pathway for the conversion it into the carbon dioxide. First step is the preparatory phase, which involves pyruvate. And what is pyruvate? We know that from the glycolysis, pyruvate molecules are produced. Pyruvate is converted to acetyl CoA by an enzyme pyruvate dehydrogenase complex. In this reaction, a molecule of carbon dioxide, NADH, FADH2, GTP that is converted into the ATP, these all are generated. This pathway is important for the synthesis of non-essential amino acids. Succinyl CoA plus glycine formed in this reaction are important for the synthesis of heme, purines and pyrimidines. And we know that what is heme? It is required for the blood synthesis. Purines and pyrimidines are nitrogenous bases required for the synthesis of DNA. It provides a number of intermediates that provide regulatory factors in number of pathways. There are number of intermediates. What are the intermediates? That are the succinyl CoA, alpha ketoglutarate, citrate. These all are the intermediates of TCA cycle that are involved in the production of some other chemical compounds. Acetyl CoA, what is acetyl CoA? Acetyl CoA is a 2-carbon compound that is formed from pyruvate. First step is we know that when the glucose break down, then it is converted into the 2 molecules of pyruvate, and then that pyruvate molecules enter into the mitochondria and by the conveyor into the acetyl CoA. There are 8 steps of Krebs cycle in mitochondrial matrix. Enzymes that are involved here, I have added a slide here to show the enzymes that are involved in the TCA cycle. Citrate synthase, econitrate, that is dehydration and rehydration is occurred by this enzyme. There is shifting of hydroxyl group, that's why it is the isomerization reaction. Isocitrate dehydrogenase, ICDH, alpha ketoglutarate dehydrogenase. It is a multi-enzyme complex which consists of 3 enzymes and 5 coenzymes. 5 coenzymes are TPP, FAD, NAD, lipoic acid and coenzyme A. Succinic dehydrogenase and fumarase, these all are the enzymes that are involved in the TCA cycle, and where they are used you can see here. Here you can see that the pyruvate molecule is converted into the acetyl CoA by the help of an enzyme pyruvate dehydrogenase complex. I have another video which is related to the pyruvate dehydrogenase complex, and there is the detailed description of pyruvate dehydrogenase complex. So it is a complex system of enzymes that converts the pyruvate into the acetyl CoA, and a molecule of NADH is released. Then acetyl CoA combines with the oxaloacetate. Acetyl CoA combines with the oxaloacetate in the presence of an enzyme citrate synthase to form the citrate. Citrate is the 6-carbon compound. Then citrate is converted into the isocitrate. Isocitrate is converted to the alpha ketoglutarate. Alpha ketoglutarate converted to the succinyl CoA. Succinyl CoA is converted to the fumarate. Here fumarate is not written, it is not clearly shown in the slide, so you will write after fumarate. you will write here the succinyl CoA, you will write fumarate and then malate. Fumarate is converted to the malate, and malate is converted into the oxaloacetate. As oxaloacetate, which is used in the first step, is regenerated, that's why it is called as the catalyst for this reaction. So the enzymes we have to memorize here, that is the aconitase. First is the aconitase that converts the citrate acetyl CoA and oxaloacetate are converted into the citrate, and citrate is converted into the isocitrate with the help of enzyme aconitrase. Then isocitrate is converted into the alpha ketoglutarate with the help of an enzyme isocitrate dehydrogenase. Then alpha ketoglutarate is converted into the enzyme succinyl CoA with the help of enzyme alpha ketoglutarate dehydrogenase. And in the conversion of isocitrate to alpha ketoglutarate, there is the removal of NAD. NAD is converted into the NADH, and there is a removal of carbon dioxide. Similarly, from alpha ketoglutarate to succinyl CoA preparation, there is also a removal of carbon dioxide and the preparation of NADH. So these are the steps. Then succinyl CoA is converted into the fumarate, and I have written the enzymes involved here in the previous slide. And then fumarate is converted into the malate with the help of enzyme fumarase, and then malate is converted into the oxaloacetate. Oxaloacetate is a 4-carbon compound. So how it is converted into the 4-carbon compound? Because we know that citrate was a 6-carbon compound. It is converted to the isocitrate, and from isocitrate to alpha ketoglutarate, one molecule of carbon dioxide is released. So it becomes 5-carbon compound. So when from alpha ketoglutarate it becomes succinyl CoA, it becomes, there is also a removal of carbon dioxide, so it becomes 4-carbon compound. So oxaloacetate, which is the 4-carbon compound, is again regenerated, and again here the molecule of NADH is produced. So that was the Kreb cycle in detail. Now we will see that by succinyl CoA there is a direct production of ATP. That is why it is called as substrate level phosphorylation, because without undergoing electron transport chain. As I have explained in the electron transport chain, as you will see in my lecture of electron transport chain, that whenever ATP is produced through electron transport chain, that is known as oxidative phosphorylation. But whenever a molecule convert itself into another molecule by the release of ATP, when ATP is generated, when a molecule is converted from one compound to the other compound, a molecule is converted into from one molecule to the other, and a molecule of ATP is generated, or 2 molecules of ATP are generated, whatever is the amount. But whenever the direct synthesis of ATP occurs, it is known as substrate level phosphorylation. But when there is the synthesis of ATP in the electron transport chain, it is known as oxidative phosphorylation. So it is a difference between oxidative and substrate level phosphorylation. So here substrate level phosphorylation occurs, and we know that one molecule of NADH produces 2.5 ATP. This is its bioenergetics, and here we can see that how many molecules of NADH are generated here. A molecule of NADH is generated here, also is generated here, also it is generated. So 3 molecules of ATP are generated here. So 3 molecules of NADH are generated here. So we know that one molecule of NADH produces NADH produces how much ATP? 2.5 ATP. So as 3 molecules of NADH are produced, so total of 7.5 ATPs are produced. 1 FADH is equal to the 1.5 ATP, and also there is 1 molecule of ATP is produced, that is produced directly from GTP synthesis. So total is the amount of 10 ATPs are totally synthesized in one TCA cycle, and we know that from one glucose molecule 2 pyruvate molecules are formed. So per molecule of glucose, 2 Kreb cycles occur. So total is the 20 molecules of ATP per glucose degradation. But for one TCA cycle, there is the production of 10 ATPs. So here is the thing, 3 NADH, 1 FADH2, 1 GTP, 2 carbon dioxide. But we know that 2 pyruvate molecules. So total is the 6 NADH, 2 FADH2, 2 GTP, that is the 2 GTP and 2 carbon dioxide. 1 GTP produces 1 ATP, so it is equivalent to ATP. So and 4 carbon dioxide, and we know that 2 carbon dioxide is released, that's why 6 carbon compound is converted into the 4 carbon compound. Here is a thing that 6 carbon compound is converted into the, here it is a 6 carbon compound, is a 6 carbon, but here a molecule of carbon dioxide is released, here also is released, so 2 carbons are removed, so the it is the 4 carbon compound that is formed at the end. So there is a molecule removal of, there is a molecules of removal, there are 2 molecular removal of carbon dioxide, 2 molecules of carbon dioxide are released. Next, the inhibitors of Kreb cycle. Fluoroacetate inhibits aconitase, arsenate inhibits alpha ketoglutarate dehydrogenase, and malonate dehydrogenase inhibits the succinic coenzyme A. Then is the regulation, it depends on the availability of the substrate. ATP is an important regulator. It is regulated by the citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. When these enzymes are available, then this process is regulated. This pathway produces ATPs, so ATP inhibits this pathway. Whenever in our body a large amount of ATP is present, then that inhibits this pathway, and NADH also inhibits this pathway, because our body will have a signal that we already have enough ATPs and NADH that can produce an ATP, so there is no requirement for the TCA cycle, and excess glucose is stored in our body, and if the glucose is converted into the pyruvate, then pyruvate is used to produce some other molecules in our body, or it is used for the again regeneration of the glucose, that is the gluconeogenesis, and then glucose is stored in our liver, or pyruvate is used to produce some other compound, that is the different compounds in our body, so it does not occur in the TCA cycle for further heat generation. Succinyl CoA inhibits alpha ketoglutarate, acetyl CoA inhibits pyruvate dehydrogenase. So we should know that which things enhance which things, and which things inhibit particular things. So this pathway produces ATP, so ATP inhibits this pathway, NADH also inhibits this pathway. Succinyl CoA inhibits alpha ketoglutarate. If a lot of succinyl CoA is present, then it inhibits alpha ketoglutarate. Acetyl CoA inhibits pyruvate dehydrogenase. If a large amount of acetyl CoA is produced, then it inhibits pyruvate dehydrogenase to not synthesize more acetyl CoA. And if succinyl CoA is present in excess, then it inhibits its previous compound alpha ketoglutarate to not be synthesized in the cell. Here is mnemonic that can be used to memorize this step. Citrate is Kreb starting sub for making oxaloacetate. You can write first word and can memorize, but the main thing is that you can also write down your own mnemonic. Okay, this is the thing that the mnemonic that I have added here, it is present in a lot of things I have seen, that this mnemonic is used by most persons. Most of the people use this mnemonic, but you can create your own mnemonic, and this is not to be written in the examination. This is only for your own memorization of this step. So you can write the names of the chemicals according to this, you cannot confuse with the next molecule, that which molecule is the next and then which is the next, so you can easily remember this. So you can also form your own mnemonics to remember these processes. So that was the all about the TCA cycle, which is also known as Kreb cycle. If you have any query related to this topic, then you can ask me in the comment section. Thank you very much.